gives you all the cell nuclei. Now I can get someone like Yusuf and say, can you segment that? Okay, and he did. And then another channel gives you another molecule of interest, the cytokeratin. This highlights all the tumor cells. Doesn't that look like a leaping tiger there? Although I'm afraid the situation is a lot more grim. And then there's another molecule called HER2/neu. It's a gene that's being overexpressed in this cancer. That means this patient is responsive to therapy that's targeting the HER2/neu gene by a drug known as Herceptin. So basically, one of the things we did as academics is said, okay, we were going to build a software system that'll go off and make sense of this data. So we built an algorithm that can segment all the cell nuclei. This is a surprisingly hard problem, actually, to delineate all the membranes, essentially delineate all the cells, and make measurements of all these cells. That's the essence of anatomic pathology. You're trying to get at structure. And then you can basically quantify the amount of these molecular markers, in this case HER2/neu, on within each cell. We know exactly where it lives and by how much. So we knew the amount of association of HER2/neu with each cell. And based on that, we can type these cells, and we can essentially tie the molecular markers to essentially cell types. You can do a lot of statistics based on that. So this was still an academic project at the time, and translation was kind of far from my mind. And then I went and gave a talk at a couple of different places. And one talk was a little bit interesting. I was just talking like this about this project, showing examples, and there was a young lady sitting in the back of the room. She's now become a wonderful collaborator, Issei Kumiko. She's from Japan. I hope I'm not mangling her name. She was sitting there with her laptop, and at the end of my talk, she came and showed, said, it works. She had downloaded the whole thing, the whole open source tool from our website, and ran it on her data. And I was kind of like freaking out and happy at the same time. So to make a long story short, there has been a mini explosion of translational adoptions of this software. And one reason why it happened is that some of my students, people like Kedar and Yusuf and Raghav, they sat there and they did something that we academics are not comfortable doing. They actually built a software system that works. They actually debugged it, and it actually works, right? It has visualization, a GUI, it has database linkage, it has everything. And it's free now. There's no barrier to adoption. So Issei Kumiko could simply sit there at the back of the seminar room, download it, and run it. And before long, we have fresh publications coming out of this. So at the University of Pennsylvania Medical Center, basically it's being already in use for 3 different renal cell carcinoma studies. One of them is funded by an R01 that we just received. One of them was funded by Pfizer. Another one by GlaxoSmithKline. Another one is for subtyping breast cancers, and there's also being applied for thyroid cancer. And at the University of Pittsburgh Medical Center, it's being used for various kinds of organ transplantation studies. All this happened in a matter of months, so translation can sometimes happen faster than we like, or rather than can be imagined. We still have a long way to go. I think that this is where realists like Mel can get into the act, and companies can build systems, labeling kits, faster microscopes that can scan the entire slide, integrate that with the software, put them on faster GPUs and servers and so on, right? And of course, companies can also, be out there in the field and help overcome some of the non-scientific adoption barriers that we face. So anyway, to make a long story short, I think this field of so-called next generation histopathology is headed towards a lot more content being extracted out of the tissue using imaging tools and image analysis tools that see more, see better, and make more sense of the data and cast it in terms that are much closer to the molecular biology that drives all of the fundamental science here. Fortunately, there's a growing role for imaging, I think, and there's a growing role for image analysis and mosaicing and just about everything that we've been doing in census. I think that just needs to happen on even more fast forward, I think.
I'd like to key off a couple of things that Audrey mentioned or alluded to. First of all, Imaging systems must be capable of imaging intact cells or intact tissue. He mentioned flow cytometry and even imaging flow cytometry, and that's a kind of a heritage that the company that I'm with, CompuCyte, grew out of. But we're very much an imaging company and recognize the need to image intact structures. And the second thing is imaging systems must be capable of molecular specificity and sensitivity. They must be capable of really quantifying the amount of, as Badri said, the molecular player involved. And that's something that flow cytometry has traditionally done very well, and that's an attribute that needs to be a part of imaging. So if we look at what it is that one can do with imaging, I've broken it down a little bit here into morphological quantification and then molecular quantification. And again, as Badri said, molecular quantification can span kind of a range between positive and negative all the way through to continuous scoring over a broad range. Okay, typically manual scoring involves very few positions on a rather limited scale. So digital pathology systems or imaging pathology systems, they emulate the manual process, but they have the opportunity or the capability in a lot of cases to go beyond that manual process. I just want to walk through a little bit of the kind of generic workflow for a typical imaging pathology system. The first, of course, is the data acquisition, the imaging, and this is done by systems in a variety of ways. Some are imaging the entire sample at high resolution. Some are imaging the entire sample at low resolution, and then regions of interest at high resolution. A couple of examples here. One is a bright field example from the genetics aerial on a tissue microarray. Tissue microarrays are essentially cores of tissue that are arrayed and then microtomed very thin and placed on a slide similar to what a tissue section would be prepared. So that's one example. Another is on a tissue section. This is rat pancreas tissue, where this sample has been imaged at low resolution, and then that low resolution image has been used to define regions of interest for further imaging at high resolution. Then the unmixing issue that Badri touched on, that too has to happen in order to really quantify molecular players. Here is actually a similar example to what Badri showed. This is a HER2-positive breast cancer tissue section, human breast tissue, stained with HER2 developed with chromatic stain DAB, and then counterstained with a blue stain hematoxylin that stains the nuclei. And you can see from the as acquired images in the center, there's really quite a bit of bleed over, if you will, from the opposing dye. And systems need to be able to separate those dyes out in order to get a true measure of the presence and the amount of the particular protein in question. Okay, then in order to determine which areas really need to be focused in on for detailed analysis, there are a number of methods. I'm showing here the new suite by Definians, where they employ an interactive learning mode, where they define certain areas of the tissue of interest and paint those areas, and then those are essentially prototypes for the other areas in that tissue and other tissues having similar characteristics or attributes. The ISIS system on the left here has the capability of either defining regions of interest manually by observing the low resolution image, or based upon expression level of antigens of interest, or a combination of that and other criteria, morphological criteria, to define those regions automatically based on a predefined protocol. Then once the images are acquired, the area of interest is identified. then there are a number of ways to extract data, but they typically revolve around segmenting the image. I'm showing here segmentation based on the Genentech aerial, where the cells are essentially color-coded in the image to identify their classification, and then that data is put into a tabular form, both the image data as well as the extracted numerical data. On the right, I show 4 different types of contouring of different antigens based on different criteria done simultaneously, and then that data is displayed in a scattergram, and the scattergram kind of portrays the flow cytometric origin of this system, the ISIS, where one can then isolate or identify and segregate populations of interest based on the bivariate plots or a series of bivariate plots. And then verifying the classification or the identification. Here again, the Genentech system, the tabulated data and the image right in the table with it. The ISIS system, the color coding that you see overlaying the image of it's actually an islet cell in rat pancreas. The overlaid color is generated by a series of identified populations in bivariate plots.